Okay, we can see your screen.
So,
we can hear you.
Right. So, hello everyone. I am Vishal. I am going to present this, and these are my co-authors, Dr. Rajalakshmi and Dr. Deborah, and I am from the team MMSS and CE. So, I will move on to the key takeaways. So, the main takeaway which we found out is that large vision language models can be efficiently adapted to these tasks just using LoRA without needing full-scale fine-tuning. So, one thing we noticed was that we could greatly save upon computational resources and also time. So, using LoRA was one thing. Other than that, we also checked on the differences between all these models, blip, Florence, and Polygamma. And we identified that cross-attention based models like Polygamma perform better than self-encoder models like blip and Florence. And something which I tried while I was doing this task was to make sure visual grounding was present properly. So I added some noise segmented images, random images with these questions to make sure the models did not depend just alone on the text, but also relied on the images, and so there are no language biases or hallucinations. So motivation and related work. Medical VQA suffers from hallucination and weak visual grounding. That was one part. And full fine-tuning needs high compute. So these are the two aspects which I focused on. So related work. So the intuition behind choosing these models are as follows. Blip was one of the first few models which were introduced for general VQA. So we chose that. And then many people were suggesting Microsoft's Florence too. So by that I chose that. And we also wanted to check a difference between the different architecture styles and Polygamma. So two reasons. One, while Blip and Florence, the parameters were in 100 millions, Polygamma had around 3 billion, I think. So we chose this. And another thing was the difference in architecture. And obviously, this was also in the baseline notebook which was provided. So I thought I could try it out. And then LoRA. So I checked upon many fine-tuning methods. So LoRA seemed the most simplest and required less computational resources. So we went for LoRA. And our main approach. So first, I tried just using Salesforce Blip model, and here I did not use any LoRA or any other methods. I just did full fine-tuning on 8K samples. So how I got the samples are as follows. I just identified 1,000 images using their IDs, and for each images, I took their respective random 10 questions for each, and I divided them into 80-20 ratio to test and train. This was done so that the data set was proper. Like for example, if I trained on the first 10,000 alone and I randomly tested it on, that's a probability that the model has never before seen these questions or images. Like maybe the first few could be only on polyps and instruments, and the others could be very different. So I made sure to there are some relation between the train and the test, even though we only chose 10K data points. And then after that, I tried it I put it in the leaderboard, and apparently the results were extremely bad. So we then tried to use LoRA to make sure we could train on the whole data set and also a bit faster. So these are the three models which we compared: Florence, Blip, and Polygamma. And visual grounding, random image question mismatches, noise segmentations. One issue which I saw in Blip and even sometimes in Florence was that after a little training, not too much, but after some samples, when the questions were asked, the answers were correct, but then it did not depend on the image. For example, if the same question was given with another random image, the answer was the same for the question, but it did not depend on the image. So I had to make sure some visual grounding was there. This was solved by the architectural change from these two and this itself. So Polygamma sort of handle it automatically. So after doing all this, I decided Polygamma would be the best both empirical and theoretical fit, because I tried 10K samples on all three of these, and Polygamma turned out to be the best empirical fit. And due to the different style of architecture, Polygamma is newer than these two and also a larger model. So I chose Polygamma to be the final one. And these are our results. So as you can see here, both the full set and the private set, the results are in so different. So we were happy that there wasn't overfitting or anything, and it's a generalized model. And as you can see in the graph, so one thing I noticed in the results was that the model performed well on presence versus absence questions. For example, box artifact present, unknown be present, and those types of question it answered perfectly. But let's say location, color, size, those questions it struggled a lot, as we can see in both of the graphs. So maybe the model lacks a bit in being more descriptive. So polyp count was one exception, but other than that, polyp size or abnormality, color, location, those aspects it struggled, but detection it did really well. So this is a small analysis of the result. And complexity wise as well, it did not differ hugely except this one part. But other than that, it was somewhat equivalent for all of them. And the lessons learned, yeah, LoRA enables scalable training under resource limits and cross attention, which we do both empirically and practically. And small data experiments can guide large scale training. These were our and some challenges. Full fine tuning caused hallucinations. Those grounding issues were still there. And also, I noticed, especially with blue metric, that medically valid answers which did not, was a proper text form. For example, during the initial stages, the answer came as polyp present. So even though that was technically correct, the score was pretty low because the given answer was a huge sentence and this was just 2 words. So that was one issue which I wanted to address. So maybe that's on the metric, or maybe the text generation part of the model is somewhat bad. And Outlook, to be frank, I saw the first presentation slide and I was like, oh my god, these two slides are very similar, like the one which shown 10 minutes ago. So replace static LoRa with task adaptive LoRa. So instead of retraining for every different task, like instead of endoscopy, instead of maybe polyp, and there could be many different. So instead of static LoRa, standard LoRa, we could use task adaptive LoRa. That was one thing which I thought I could try. And then some sort of RAG, like there can be a knowledge base to make sure the answers are, factually correct instead of just training on these models. If there was some knowledge base like UMLS or SNOME, then these could help a lot in validating the model's accuracy and calibrate output confidence using temperature scaling and Monte Carlo dropout. So to make sure neuron dropouts were there and like how different the answers gave, how different the answers were when some sort of dropouts were there, and scaling to make sure the probabilities were right. Like it doesn't, give overconfident metrics. And then grounding maps, grad cam over image tokens and bounding box. So I'm sure these were addressed in Subtask 2. I only did Subtask 1. So, and move beyond these metrics. And one thing which I just which I had noticed earlier as well, the score similarities between my team and the other team. I noticed that in the public report as well. So what I did was basically use the baseline notebook 95% without any changes at the end, because Polygamma proved to be right and the model parameters. So maybe if that team used the same model as well, maybe there's a chance that the results are the same. But I noticed this in the public leaderboard as well, and I submitted second, so I was scared. But yeah,